Today we are going to discuss on the topic oxidative phosphorylation. The process of oxidative phosphorylation is closely associated with the functioning of the electron transport chain. In oxidative phosphorylation, ATP is produced by combining ADP and inorganic phosphate with the energy generated by the flow of electrons from NADH to molecular oxygen in the electron transport chain. There are three sites in the respiratory chain where ATP is formed by oxidative phosphorylation. Since hydrolysis of ATP to ADP and inorganic phosphate releases around 7.3 kilocalories per mole, the formation of ATP from ADP and inorganic phosphate requires a minimum of around 8 kilocalories per mole. There are three sites in the respiratory chain where ATP can be formed. Site 1 involves the transfer of electrons from NADH to CoQ. Obviously, this step is omitted by succinic dehydrogenase, whose FADH2 prosthetic group transfers its electron directly to CoQ bypassing NADH. Site 2 involves the transfer of electrons from cytochrome B to cytochrome C1 and then it is transferred to cytochrome C. Site 3 transfer electrons from cytochrome A3 to molecular oxygen, which is blocked by carbon monoxide, cyanide, hydrogen sulfide and azide. Let me explain the mechanism of oxidative phosphorylation. Three major proposals for the mechanism of oxidative phosphorylation have been considered. The synthesis of ATP is carried out by a molecular assembly in the inner mitochondrial membrane. This enzyme complex is called mitochondrial ATPase or H plus ATPase. It is also called ATP synthase. Much information is now available regarding H plus ATPase, which consists of a head piece, a stalk and a base piece. The stalk serves as the proton channel. The head piece protein assembly is called F1 unit. F1 protrudes from the inner surface of the plasma membrane. F1 is thought to be alpha 3, beta 3, gamma, delta, epsilon. The base piece consists of transmembrane integral protein called FO unit. FO is thought to be alpha 1, beta 2, C 10 to 12. The F1 subunit functions as an ATPase, while F1 unit, when bound to FO, serves as proton translocating ATP synthase and catalyzes phosphorylation of ADP to ATP. The three hypotheses do make use of the information available on ATP synthase. The three theories on oxidative phosphorylation were the chemical coupling hypothesis. This is developed from the concept of high energy intermediate common to both electron transport and phosphorylation of ADP. The second theory is the conformational coupling hypothesis. According to this hypothesis, the mitochondrial cristae undergo conformational changes, and these changes in architecture of the mitochondrial cristae reflect the changes in the different components of the electron chain to one another. It is believed that these conformational change represents the formation of high energy state. The most accepted view of oxidative phosphorylation is the third theory, chemiosmotic coupling hypothesis, which was postulated by Peter Mitchell in 1961. He proposed that electron transport and ATP synthesis are coupled by a proton gradient across the inner mitochondrial membrane rather than by a covalent high energy intermediate or an activated protein conformation. He postulated that proton motive force drives the synthesis of ATP by the ATPase complex. The central idea is the energy capture related to the movement of protons across the inner mitochondrial membrane, which serves as the integral part of phosphorylation mechanisms. Transport of electrons in the electron transport chain causes pumping out of H+ across the inner membrane into the matrix to create a H+ gradient. The pH outside is 1.4 units lower than inside, and the membrane potential is 0.14 volts, the outside being positive. In the electron transport chain, H+ is separated from the electron. Thus, as the electrons move down the chain, H+ is expelled, traveling from the matrix to the intermembrane space. The protons in the intermembrane space pass through the inner membrane and back into matrix at a special site or pore where ATP synthase is located. The dissipation of energy that occurs as the protons pass down the concentration gradient to the matrix allows the phosphorylation of ADP by ATP synthase. Thus, high energy compound ATP molecules are formed with the help of oxidative phosphorylation. Inhibitors of oxidative phosphorylation: oligomycin. This prevents the stimulation of oxygen uptake by ADP and phosphorylation of ADP to ATP. Atractyloside. It is a toxic glycoside. It blocks the translocase that is responsible for the movement of ADP and ATP across the inner mitochondrial membrane. Bongregate is another inhibitor of oxidative phosphorylation, which is a toxin produced by pseudomonads and acts very similar to atractyloside. Uncouplers of oxidative phosphorylation. Uncouplers are the compounds that allow mitochondria to use oxygen regardless of whether or not there is any phosphate available. When an uncoupler is added, there is marked increase in oxygen uptake. The uncouplers are 2,4-dinitrophenol, which is a classic uncoupler of oxidative phosphorylation. Another uncoupler is dicumarol, which is used as an anticoagulant, also acts as an uncoupler. Calcium acts as an uncoupler. Transport of calcium ion into mitochondria can cause uncoupling. When calcium is transported into mitochondria, electron transport can proceed but energy is required to pump the calcium into the mitochondria. Hence, no energy is stored as ATP. The next uncoupler is CCCP, that is chlorocarbonyl cyanide phenyl hydrazone. This is the most active uncoupler. The next active uncoupler is valinomycin. This is produced by a type of Streptomyces. This transports potassium from the cytosol into the matrix and H+ from matrix to cytosol, thereby decreasing the proton gradient. Other physiological uncouplers are excessive thyroxine hormone, long-chain fatty acids in brown adipose tissue, and unconjugated hyperbilirubinemia.